Chapter 2. Exploring Substances, Acidic, Basic, and Neutral. On the 28th of February, the school hosted a science fair to celebrate National Science Day. At the entry gate, siblings Ashwin and Kirthi were greeted with a white sheet of paper. They were curious to know why a white sheet of paper was given to them. A few steps ahead, there was a volunteer spraying a liquid on these sheets of paper. The siblings also got their white sheets sprayed. To their surprise, the words, "Welcome to the wonderful world of science," appeared on the papers the moment the liquid was sprayed. Figm 2.1. They were excited and eager to know how this happened and the reason behind it. Their curiosity was partly satisfied at the colorful world of substances stall. They saw many activities showing color changes on mixing different substances. They decided to explore these changes further. Let us join them on this learning adventure. Number 2.1, Nature. Our science laboratory. Tabs 2.1.1, Litmus as an indicator. Activity 2.1, let us explore. Collect samples of lemon juice, soap solution, amla juice, tamarind water, vinegar, baking soda solution, tap water, washing powder solution, sugar solution, and salt solution. Take a strip of blue litmus paper and cut it into small pieces. Spread these pieces on a clean and dry white tile. Using a dropper, put one drop of each sample, one by one, on the blue litmus paper pieces, as shown in 2.2a. Fig 2.a, color change in blue litmus paper. Do you observe any change in the color of the blue litmus pieces? Record your observations in Table 2.1. Repeat the same activity with pieces of red litmus paper, as shown in Fig 2.2b, and record your observations in Table 2.1. Fig, color change in red litmus paper. How to prepare lime water. Do not confuse lime water with the word lime, which is a fruit similar to lemon. Lime water, solution of calcium hydroxide in water, can be easily prepared by mixing lime, i.e. calcium oxide, in water and leaving it undisturbed for some time, say 1 hour. Filter the liquid into another container and use it as lime water. Table 2.1, testing the nature of samples with blue and red litmus papers. SNO, name of the sample. 1. Lemon juice. 2. Soap solution. Amla juice. 4. Tamarind water. 5. Vinegar. 6. Baking soda solution. 7. Lime water. 8. Tap water. 9. Washing powder solution. 10. Sugar solution. 11. Salt solution. 12. Any other. The table has 2 blank columns for students to fill in. Color of blue litmus paper after putting a drop of sample. Color of red litmus paper after putting a drop of sample. Text below the table. Now let us analyze Table 2.1 and sort the samples into 3 groups as follows. Group A, samples that turn the blue litmus paper to red. Group B, samples that turn the red litmus paper to blue. Group C, samples that do not affect either of the 2 litmus papers. Record the data in Table 2.2. Table 2.2, grouping of samples tested in Table 2.1. Fig 2.3, blue and red litmus paper strips. A student asks, open quote, "I am curious to know, what are these red and blue litmus paper strips made of? Why do they change color when drops of some samples are put on them?" Close quote. Let us find out. Litmus is a natural substance obtained from lichens. It is available both as a solution and in the form of paper strips, known as litmus paper. The litmus paper is available in 2 colors, blue and red, as shown in Fig 2.3. Substances that turn blue litmus paper to red are acidic in nature, while those that turn red litmus paper to blue are basic in nature. Since litmus shows different colors in acidic and basic solutions, it is called an acid-base indicator. Some other substances, both natural and synthetic, can also be used as indicators. Synthetic indicators are made in laboratories, and you will learn more about them in higher grades. Holistic lens lichens are formed by the association of 2 living organisms, a fungus and an alga. They grow on rocks and trees in regions that have abundant rainfall and clean air. Do you find lichens on trees in your neighborhood? Now let us classify the substances sorted in Group A, Group B, and Group C in Table 2.2. The substances in Group A, such as lemon juice, amla juice, tamarind water, and vinegar, turn the blue litmus paper to red, implying that these substances are acidic in nature. The substances in Group B, such as soap solution, baking soda solution, lime water, and washing powder solution, turn the red litmus paper to blue. Hence, these substances are basic in nature. The substances in Group C, such as tap water, sugar solution, and salt solution, did not change the color of either litmus paper. Can you predict their nature? These substances are said to be neutral because they are neither acidic nor basic. Activity 2.2. Let us relate and explore. Are all the substances in Group A of Table 2.2 edible? Have you ever tasted these edible substances? Can you recall their taste? You will find that all these substances taste sour. Thus, we can say that substances that taste sour tend to contain acids and are acidic in nature. Caution: do not taste anything until asked to do so. Do not taste any unknown substance. Some common edible substances and the names of the most common acids present in them are given in Fig. 2.4. Fig. 2.4. The most common acids present in some edible substances: orange, citric acid; amla, Indian gooseberry; ascorbic acid, vitamin C; and citric acid, tamarind; tartaric acid, tomato; citric acid, and oxalic acid. Find out and write the names of the most common acids present in the following substances: lemon, curd, tamarind, vinegar. Now let us take one of the substances, baking soda solution, from Group B. Rub the baking soda solution between your fingers. What do you observe? It feels soapy or slippery. Basic substances are generally slippery to touch. Also, bases generally taste bitter, but everything that tastes bitter may not contain a base. For example, bitter gourd, Corella, possesses a bitter taste but is not basic in nature. A student says, open quote, "Now I can check the nature of floor cleaning liquid using an indicator," close quote. Another student asks, open quote, "If litmus is not available, are there some other natural substances that can serve as acid-base indicators?" Number 2.1.2, red rose as an indicator. You might have observed many colored flowers in your surroundings. Try making your own indicators using these flowers. Activity 2.3, let us prepare. Collect some fallen petals of red roses available in your surroundings, Figure 2.5. It is advised not to pluck flowers. You may pick petals or flowers fallen on the ground. Take a fistful of the collected petals of red roses and wash them with water. Crush the petals using a mortar and pestle. Place them in a glass tumbler. Pour some hot water into the glass tumbler so that the crushed flower petals are completely immersed. Caution: perform this step under the supervision of an adult. Cover the glass tumbler with a lid. Wait for 5 to 10 minutes until the water becomes colored, Fig. 2.6, and filter it. The filtrate, liquid after filtration, is the required flower extract, Figure 2.7, to be used as an acid-base indicator. Figures: Fig. 2.5, red roses; Fig. 2.6, red rose petals immersed in hot water; Fig. 2.7, test tube containing the red rose extract. Activity 2.4, let us find out. Place 10 to 20 drops of the prepared red rose extract in each of 2 small transparent bottles or test tubes. Mark them A and B. Add 20 to 30 drops of lemon juice in test tube A and 20 to 30 drops of soap solution in test tube B with the help of droppers. Observe and record any color changes, Fig. 2.8, to the extract in Table 2.3. Repeat the same with the other samples used in Activity 2.1 and record your observations in Table 2.3. Fig. 2.8, the changes in color of the red rose extract on adding lemon juice A and soap solution B. Table 2.3, testing the nature of samples with the red rose extract. No. Name of the sample. The color of the red rose extract after adding the sample. Nature of the substance. 4. 1. Lemon juice. 2. Soap solution. 3. Amla juice. Discuss your observations with your classmates. Are the samples that change the color of the flower extract to a shade of red the same as those that change the color of blue litmus paper to red? Group A, Table 2.2. Are the samples that change the color of the flower extract to a shade of green the same as those that change the color of red litmus paper to blue? Group B, Table 2.2. Are the samples that do not change the color of the flower extract the same as those that did not change the color of red and blue litmus papers? Group C, Table 2.2. Conclusion. From the above activity, we can conclude that the red rose extract can also be used to test the nature of substances. Hence, it is another example of an acid-base indicator. We can conclude that the red rose extract gives red color in an acidic solution, green color in a basic solution. Can you now fill in the nature of the substances in Table 2.3? We are sure that you will be excited about the above results. You may repeat the process of preparing the extract and testing substances with some vegetables, fruits, or flowers, such as beetroot, purple cabbage, turmeric, Indian blackberry, jamun, and red hibiscus, Goodhall, flower. They can also act as acid-base indicators. Fascinating facts. Hydrangea is a plant that grows in cooler climates in the Himalayan region and the northeastern states. It gives flowers of different colors depending on the nature of the soil. Acidic soil produces blue-colored flowers. Basic soil produces pink or red flowers. Can gardeners alter the color of hydrangea flowers by adjusting the acidic or basic nature of the soil? Number 2.1.3 Turmeric as an indicator. We have used blue and red litmus paper strips in Activity 2.1. Can you also make paper strips with some other natural indicators? Find out by performing the following activity. Activity 2.5. Let us prepare. Take a spoonful of turmeric haldi in a petri dish or container and add a little water to make a paste, Fig 2.9a. You may also grind a piece of fresh turmeric. Carefully dip a piece of filter paper in the turmeric paste until it gets yellow color. Take it out and allow it to dry. Cut this yellow paper into thin strips, which are used as turmeric paper, Fig 2.9b. Caution: perform this step under the supervision of an adult. Fig 2.9, preparing turmeric paper. A. Turmeric paste. B. Turmeric paper strips.